Welcome to the deep dive, where we take complex information and pull out the nuggets that really matter, giving you a smart shortcut to understanding the world around you. You're here because you want to learn to get those aha moments without feeling buried under jargon and endless details.
Exactly. And today we're embarking on a truly exciting exploration into the cutting edge of scientific discovery. We've got a fascinating research paper titled Robin, a multi-agent system for automating scientific discovery that's packed with potential.
Okay, Robin, a multi-agent system automating scientific discovery. Wow, that sounds incredibly ambitious. So what exactly is Robin, and, why is it such a big deal?
Well, at its core, Robin is an artificial intelligence system. It's designed to mimic the entire intellectual process of scientific discovery, all within a single unified workflow. Think of it like assembling a dream research team, where each member or agent has a specific job and they all work together seamlessly.
An AI dream team.
Okay.
And this team actually achieved something significant, right? Something related to a major health challenge.
Absolutely. A really big one. The researchers used Robin to tackle dry age-related macular degeneration, or DAMD. That's a leading cause of irreversible blindness. And remarkably, Robin identified a completely new potential way to treat this condition.
Wow. A new way. For DMD, that affects so many people, and yeah, current treatments are pretty limited. So Robin didn't just point to a potential drug, it sounds like. It also started to figure out how it might work. It's a much deeper level of insight.
Precisely. That's what's so exciting. The paper highlights that Robin is the first system to automate all those crucial intellectual steps. From the initial background research to forming hypotheses, designing experiments, and analyzing the results, all in one continuous loop.
Those intellectual steps. It's more than just like searching a database, I imagine. What does that iterative process look like for Robin?
Yeah, much more. Imagine a scientist tackling a new problem. They'd first dive into the existing research, right? Then formulate hypotheses, possible explanations. Next, they design experiments to test those ideas, analyze the data, and, based on those findings, they might refine their initial ideas or come up with new ones. Robin automates this whole cycle.
So it's like having a research partner that can just tirelessly explore this vast landscape of scientific knowledge and make connections maybe we'd miss.
In a sense, yes.
Right.
Exactly. Robin incorporates different AI agents, each with a specific role. You've got Crow and Falcon. They're experts in sifting through scientific literature. Crow is like the fast scout, quickly identifying key info for strategies and potential treatments. Falcon then does the deep dive, thoroughly evaluating the most promising candidates,
Right.
like a meticulous detective maybe. And then there's Finch. Finch is the data whiz. It autonomously analyzes the results from the lab experiments.
Okay, so it's not just passively reading, though. Robin is actively, thinking, proposing things, interpreting.
It's exactly right. It operates semi-autonomously. It generates its own hypotheses about how a disease might work or how it could be treated. It then proposes specific experiments.
Yeah.
When the experimental data comes back from human researchers in the lab, mind you, Finch analyzes it. And then Robin uses those findings to update its understanding, maybe generate new, more refined hypotheses. It's this constant cycle of learning and discovery.
And the paper actually says Robin produced all the hypotheses, experimental plans, data analyses, even the figures in the paper itself.
It does, which really underscores the level of autonomy we're talking about here.
Okay. Wow.
And to really appreciate why this matters, let's think about why scientific discovery, especially in drug development, can be, so slow and challenging sometimes. Consider drug repurposing, finding new uses for existing drugs.
Right. Drug repurposing. That seems like it should be faster, using drugs we already know are safe for something else.
It can be, but history shows us that even with existing drugs, finding those new uses can take a surprisingly long time. The paper gives examples like dabrafenib, a cancer drug. Its basic action was understood around 2010, but its potential to help prevent hearing loss wasn't found until a decade later, just through broad, human-led screening.
A decade! Wow. That's a huge delay for people who could have benefited. What causes those kinds of lags? Is it just too much information?
Often, yeah. It comes down to the sheer volume and complexity of scientific information. It's incredibly difficult for human researchers to keep track of and connect all the seemingly unrelated pieces of knowledge, pieces that might point toward a new use for a drug, and imagine a massive jigsaw puzzle, pieces scattered across thousands of publications.
Yeah, I can see that. And this is where AI, especially these large language models like the ones inside Robin, can really make a difference, right? They can process and recall way more data than any human possibly could.
Precisely. These models are trained on enormous data sets. It allows them to spot patterns and relationships across diverse scientific fields. Connections a human might just never see because of the scale. This ability to connect seemingly disparate ideas is absolutely key to speeding things up.
Okay, but we've seen AI used for suggesting hypotheses before. What really sets Robin apart from those earlier systems?
That's a really critical distinction. While older AI systems might have focused on, say, just suggesting hypotheses or automating one specific step, Robin is the first to fully integrate the entire intellectual workflow. It doesn't just stop at suggesting an idea. It goes on to figure out how to test that idea, analyzes the data that comes back, and then uses that data to refine its understanding and generate new hypotheses. It's a continuous, self-improving loop, an end-to-end automated scientific reasoning system, really.
Right, the whole process. Okay, let's dive into the specifics then. How does Robin actually operate day to day, so to speak? We know Crow and Falcon handle literature, Finch handles data. Can you elaborate a bit?
Sure. Think of Crow as the rapid intelligence gatherer, quick scans of a wide range of literature, identifies potential strategies, promising therapeutic candidates, quick leads. Falcon then acts as the in-depth analyst, takes those leads from Crow, conducts really thorough investigations. It generates detailed reports evaluating each candidate based on all the available evidence.
Okay, so Crow flags possibilities, Falcon does the serious vetting, and Finch takes over once there's actual experimental data to look at.
Exactly. Finch is the autonomous data analyst. Once the human scientists run the experiments in the lab, the raw data gets fed to Finch. Finch analyzes it, determines the outcomes, and that analysis then informs the next steps in Robin's iterative cycle, maybe leading to new hypotheses, maybe refining old ones, guided by Falcon's deep understanding of the literature and now the new data.
It's a closed-loop system then, constantly learning and refining. But the paper mentions human scientists are still involved in the lab work, right? So it's a collaboration.
Yes, absolutely. It's a synergistic partnership. Robin does the heavy lifting of knowledge synthesis, hypothesis generation, data analysis, while human researchers contribute their essential expertise in designing and physically doing the lab experiments, providing that crucial real-world data.
Fascinating. Okay, now let's focus on how Robin actually tackled dry, age-related macular degeneration. Why DMD? Why was that chosen as the proving ground for this system?
Several factors made DMD a really good test case. First, it's a major global health problem growing too. It's the leading cause of irreversible vision loss in developed nations. Second, despite how common and serious it is, current treatment options for the dry form are very limited. So there's a huge unmet medical need, a strong incentive for new ideas.
Right. A widespread problem with few good answers makes sense as a target. So what were Robin's first steps when it started looking into DMD?
It began by reviewing a pretty substantial body of existing research, 151 relevant papers on DMD. From that initial broad survey, it was able to propose 10 different biological mechanisms, things believed to contribute to the disease developing or getting worse.
10 potential mechanisms. How did Robin decide which one of those to, focus on?
It used an internal evaluation process,
Mm-hmm.
ranked them based on factors like the strength of the evidence in the literature, biological plausibility, things like that.
Mm-hmm.
Ultimately, it zeroed in on enhancing the phagocytic function of retinal pigment epithelium cells, or RPE cells, as a particularly promising therapeutic strategy.
RPE cells and phagocytosis. Okay, could you maybe just briefly explain that connection to DMD for listeners who aren't deep into retinal biology?
Sure thing. RPE cells are vital for keeping the retina healthy. One of their key jobs is phagocytosis, basically. It's the process of engulfing and clearing out waste products that build up in the retina naturally. In DMD, this cleanup process becomes less efficient. Debris builds up, damages the photoreceptor cells, the light-sensing cells, and that ultimately causes vision loss. So Robin's logic was, if we can boost this cellular cleanup mechanism, maybe we can fight the disease.
Okay, that makes the strategy clear. Boost the cellular waste removal. So Robin picked that strategy. What was the next move?
Right. Once it had the strategy, Robin did a much more focused literature review. Looked at around 400 papers specifically related to RPE phagocytosis and existing drugs known or suspected to influence this process. This led to a list of 30 existing drug candidates that Robin proposed testing in an assay designed to measure how well they enhance that RPE phagocytic capacity.
30 potential drugs. It's still quite a few to check out. How did Robin narrow that list down for lab testing?
That's where Falcon's detailed analysis really came in handy. For each of those 30 candidates, Falcon generated these comprehensive reports, looking at the scientific rationale, why the drug might work, its known properties, the strength of the evidence, all of it. These reports were then used in another internal ranking process to prioritize the absolute most promising candidates for that first round of experimental validation.
And which drugs actually made it to the top of that list for the initial tests?
The top 5 selected for that first round of lab experiments were Exendin-4, fingolimod, MFGE8, Y27632, and a combination of AI-CAR and 2DCA.
Okay, so those were the first ones put to the test in the lab. What did those experiments actually involve? What did they look like?
They used a cell-based assay, so RPE cells in a dish were treated with each of these drug candidates. Then, tiny fluorescently labeled beads were added. The idea was to measure how effectively the RPE cells could engulf these beads that indicates their phagocytic activity level. This engulfment was then quantified using flow cytometry, a technique that lets you analyze individual cells based on their fluorescence.
And this is where Finch, the AI data analyst, took over the reins.
Exactly. The raw data from the flow cytometry experiments was fed into Robin, and Finch autonomously analyzed it. It compared the activity of the cells treated with the drugs to a control group. And interestingly, Finch's analysis confirmed that Y27632, one of those top candidates, did indeed enhance RPE phagocytosis. And this finding was actually consistent with some earlier research that Robin itself had identified during its literature review.
Got it. So one of Robin's top picks showed a positive result pretty quickly, but the investigation didn't just stop there, did it? Robin wanted to understand why Y27632 was working.
Correct. That's the next logical step in science, right? Based on those initial positive results, Robin recommended a follow-up experiment using RNA sequencing. RNA-seq is a powerful technique. It lets researchers see which genes are active being turned on or off in the RPE cells after the Y27632 treatment. Gives you a snapshot of the molecular changes.
And Finch was able to handle the analysis of that complex RNA sequencing data as well. That's pretty sophisticated stuff.
Yes, it was. Finch performed what's called a differential gene expression analysis, DGE analysis. It pinpoints genes whose activity is significantly altered by the drug. This analysis revealed that Y27632 doesn't just affect the physical process of eating the beads, but it also induces rapid changes in the gene expression patterns during phagocytosis.
So it's changing things at a deeper level. Were there any specific gene changes or pathways that really stood out from Finch's analysis?
Several key pathways popped up. Things involved in organizing the actin cytoskeleton that's crucial for the cell-changing sheep to engulf things. Also important signaling pathways, and autophagy, which is like the cell's internal recycling system.
Right.
But perhaps the most significant finding was a threefold increase in the expression of ABCA1. Yeah, it's a gene coding for a critical protein involved in transporting lipids fats out of cells. And this is particularly relevant to DMD because defects in a related protein, ABCA4, are known to be involved in some forms of macular degeneration. Plus, lipid buildup is a hallmark of DMD. And another protein, ApoE, which interacts with ABCA1, has also been linked to the disease. So it's hitting a very relevant pathway.
Wow. Okay, so Robin didn't just find a drug that works in the assay, but it also pinpointed a potential molecular mechanism directly relevant to DMD pathology. That seems like a huge step.
It really is. Understanding the why is crucial, and Robin didn't stop there. It used the knowledge gained from those Y27632 experiments to conduct another iteration of drug candidate hypothesis generation, leveraging its now updated understanding of the relevant biology.
Another cycle of learning and discovery. Okay, so what came out of the second round? Did it find something even better?
It did. After analyzing the new experimental data from testing more candidates, Finch showed that Ripasudil. Ripasudil is another drug that inhibits the same target as Y27632. It's a ROCK inhibitor. But this one is actually already approved for treating glaucoma in Japan. Finch showed that Ripasudil actually outperformed Y27632 in enhancing RPE phagocytosis. It caused a 7.5-fold increase compared to the control. Much better.
A 7.5-fold increase. That's substantial. And the fact that Ripasudil is already approved for an eye condition, glaucoma, that must give it a major advantage for repurposing for DMD, right? I mean, its safety profile for use in the eye is likely already well established.
Exactly. That's a huge plus. Ripasudil's existing clinical approval and safety record make it a very, very promising candidate for potentially rapid repurposing for DMD. What's also remarkable here is that Robin was the first to propose ROCK inhibitors for dry AMD, specifically based on this effect on phagocytosis. Previous suggestions for using ROCK inhibitors in retinal diseases had mostly focused on wet AMD and their effects on blood vessel growth. That's a different mechanism altogether.
So Robin made a novel connection. It linked ROCK inhibition and phagocytosis in the context of dry AMD, a connection human researchers hadn't explicitly made before, even though maybe the pieces of information were out there in the literature.
Precisely. It synthesized the information in a new way. The paper even notes Robin pulled together info from a study showing why 27632 could boost phagocytosis, specifically in RPE cells that had low activity to begin with, demonstrating its ability to connect these disparate findings.
This whole process just brilliantly highlights the potential of this approach to really accelerate finding new therapeutic strategies. The paper also touches on the bigger picture, the broader implications for drug discovery in general, doesn't it?
It does, yeah. The authors talk about the increasing challenges, the costs associated with traditional drug discovery, and just the pressing need for new, more efficient approaches. They present Robin as a system that can significantly speed up this process by automating those key intellectual steps and efficiently exploring just vast amounts of scientific data.
And Robin's potential isn't limited to just DMD, right? I think you mentioned it generated hypotheses for other diseases, too.
That's right. To show it wasn't just a one-off success, the research team had Robin generate therapeutic hypotheses for 10 other diseases with significant unmet medical needs, showcasing its potential across a range of different areas.
Of course, like any new technology, it probably has limitations and areas for improvement still. What are the researchers focusing on for the future development of Robin?
Sure. Several key areas are priorities. One is enhancing Robin's ability to generate really detailed, directly executable experimental protocols. That would reduce even more human interpretation in the lab setup. Another focus is improving Finch's autonomy in what they call prompt engineering, basically making it even more adaptable for analyzing diverse types of experimental data without needing as much human guidance on how to approach it. And finally, they're working on ways to better integrate and align Robin's hypothesis generation and evaluation with the nuanced judgment and, frankly, intuition of human scientists to ensure the highest quality, most relevant hypotheses come through.
Makes sense. But even at this stage, it really feels like Robin represents a significant leap forward in AI-driven scientific discovery. It's a new way of thinking about tackling these complex biological problems and speeding up treatment development.
Absolutely. By integrating hypothesis generation, experimental planning, and data analysis into this single, continuously learning system, Robin offers a powerful new paradigm that has the potential to not only revolutionize therapeutic development, but maybe fundamentally advance our understanding of the natural world across many scientific fields.
Okay, so just to recap this deep dive for everyone listening. Robin, this AI system, successfully identified an existing drug, Ripasudil, as a really promising new treatment candidate for dry, age-related macular degeneration, and it achieved this by autonomously navigating this huge landscape of scientific literature, generating and prioritizing hypotheses, proposing experiments, analyzing the results, and constantly refining its understanding.
That's the key takeaway, yes. This work demonstrates a major stride towards, truly AI-driven scientific breakthroughs, and in a critical area of unmet medical need, offering real hope for new treatments.
And it really underscores the transformative potential of this kind of technology. It could dramatically accelerate the pace of discovery across a whole spectrum of scientific fields.
Indeed. Which leads to a pretty compelling question for you to think about. As AI becomes increasingly sophisticated, increasingly capable of conducting scientific research, how might this fundamentally reshape the roles of human scientists in the future? Could it lead to entirely new types of scientific inquiries, discoveries that maybe we can't even envision today? It's a fascinating prospect to mull over.